A very good morning and selamat pagi to everyone. A warm welcome to all participants joining us from Malaysia, the SEAMEO network, and our affiliated countries. Thank you for being here for the IMR-SEAMEO Webinar Series 2025. Today's theme is Data-Driven Safety from In Vivo Studies to Pharmacovigilance Practices. Our presenters are from the Institute for Medical Research, IMR, and Hospital Shah Alam. I'm Dr. Lau Mei Siu from the Toxicology and Pharmacology Unit, Herbal Medicine Research Center, HMRC, IMR, and I will be your moderator today. At HMRC, we explore evidence-based approaches to herbal products, bridging preclinical safety and quality systems such as good laboratory practice with translation towards clinical trials. We are excited to share practical insights you can apply in your settings. So before we begin, just a few quick notes. Firstly, we won't take questions during the talks, but please post your questions using the Q&A link shared in the YouTube description. We will address them during the Q&A after both presentations. Secondly, an attendance link is provided in the YouTube description. Please fill it in to receive your e-certificate. Thirdly, do subscribe to the National Institutes of Health NIH Malaysia YouTube channel and click the notification bell to catch future sessions. We have two speakers today. First, Puan Suganthi Jeyabalan from HMRC IMR, followed by Dr. Azra Mohamad Pauzi from Hospital Shah Alam. Our first speaker is Puan Suganthi Jeyabalan, a pharmacist who serves as the head of Good Laboratory Practice (GLP) at the Herbal Medicine Research Center (IMR). She also holds the role of Deputy Test Facility Management at the Non-Clinical Research Facility (IMR). Today, she will speak on implementing OECD-GLP in clinical toxicology, a pathway to regulatory success, drawing upon her experience in the field. Puan Suganthi, the screen is yours.
Thank you very much for the warm introduction, Dr. Lau. A very good morning to all esteemed listeners. It is truly an honor to stand before you today and have the opportunity to share our expertise and experience with you. The topic that I will be presenting today is implementing OECD-GLP in preclinical toxicology, a pathway to regulatory success. Here's a brief overview of what I'll be covering in today's presentation. I will begin with a drug discovery and development pathway, followed by preclinical safety studies. Next, I will touch on OECD-GLP and provide some insights into the implementation of OECD-GLP in Malaysia. Following that, I will introduce our OECD-GLP compliant test facility, also known as NCRF-IMR. And finally, I will wrap up by highlighting some key roles and responsibilities of sponsors, along with some of real-world challenges that they often encounter when submitting their product to a GLP-compliant test facility. Let's start off with the drug discovery and development pathway. Drug discovery and development pathway is widely recognized and used globally as a reference by regulatory authorities and other stakeholders into guiding them towards evaluation and subsequently approval of health-related products. Understanding this pathway is essential for both researchers and sponsors as it highlights the critical processes required to ensure the successful registration of products, whether be it pharmaceutical products or even natural products or herbal products, but with a reminder, with therapeutic claims, of course. As shown here, this pathway begins with the discovery of a drug, such as molecule or compounds, which then progresses into the preclinical phase. During this stage, various tests are conducted to evaluate the new drug, as in the formulation, quality, efficacy, and safety before advancing into clinical trials. This is a snapshot of both the preclinical and clinical phase with a focus on the quality systems applied throughout the process. As highlighted in the red area, safety studies, also referred to as toxicity studies, must be conducted under strict good laboratory practice GLP conditions. Moving into the clinical phase, other quality systems come into play, including good manufacturing practice GMP and good clinical practice GCP. But why is it so important to even have all these quality systems in the first place? It is because by implementing robust quality systems across the stages in a drug development process, it safeguards patients' health by ensuring that only safe and high quality and effective products reach the targeted population. Okay, let's move on to preclinical safety study. Preclinical testing is a mandatory phase in a drug development process. Its primary objective is to evaluate the safety and other activities of the product, ensuring that it poses no unacceptable risk before advancing into human trials. In fact, the data generated during preclinical testing is used as a reference for justifying and designing clinical trials in human subjects. Okay, so preclinical testing is typically conducted using a combination of in vitro, as in using cell lines or assays, and also in vivo by using living organisms such as rodents and non-rodents animals. Not all types of toxicity studies are necessary. The requirement in conducting preclinical safety studies depends on the nature of the product, its intended use, and the duration of the use in humans or patients later. So here in my slide here, I have listed down the types and of toxicity studies that can be conducted. As in general toxicity, we have the dose range finding study, single dose toxicity study, and repeated dose toxicity study. You can also conduct specific toxicity studies based on the need of your product. Safety pharmacology is a testing of safety on our organ systems, the primary organ systems such as respiratory system, cardiovascular system, and central nervous system, for example. And for in vitro toxicity testing, we have the Ames test, in vitro chromosome aberration assay test, and also in vitro micronucleus assay test. Let's dive into the OECD-GLP. What is OECD? So OECD actually basically stands for Organization for Economic Cooperation and Development. It is an international organization whereby it consists of 38 member countries and 7 non-member mutual acceptance of data MAD countries. And what is GLP? GLP stands for Good Laboratory Practice. Okay, so it's a quality system whereby preclinical safety studies are planned, performed, monitored, recorded, reported, and archived. The integration of GLP standards within the OECD framework resulted in the formation of OECD-GLP 10 principles and the development of harmonized OECD testing guidelines. In a nutshell, OECD-GLP stands as the gold standard in laboratory practices, making sure the study data we produce is both trustworthy and of quality. The GLP study data generated are not only reliable and repeatable, but also are fully auditable and internationally accepted. It is important to note that OECD-GLP standards are specifically designed for preclinical toxicity testing. They do not extend to other types of preclinical studies such as efficacy and so on. Okay, so what is the standing of OECD-GLP in Malaysia? Okay, so although Malaysia is not a full member OECD country, but it is fully adhered to Mutual Acceptance of Data (MAD) system under the OECD-GLP framework since 2013. Notably, Malaysia is also the second country in Southeast Asia to achieve this recognition, reflecting its strong commitment towards international standards in laboratory data quality and integrity. According to our regulatory authority, NPRA, GLP compliance is mandatory for approval and registration of pharmaceutical and other related products in Malaysia. Okay, so now I will talk about our OECD GLP compliant facility, the NCRF IMR. We are also known by its full name, which is a Non-Clinical Research Facility, IMR. We made history on 7 February 2014 by becoming the first laboratory in Malaysia and in Southeast Asia to receive OECD GLP compliance certification in the field of toxicity studies. This milestone has placed us on the global stage of GLP compliant countries and opened up opportunities for its safety data to be accepted and entrusted internationally. NCRF IMR's continued commitment to quality and compliance is overseen by our National Pharmaceutical Regulatory Agency, NPRA, which acts as the official GLP compliance monitoring authority. Our OECD GLP certification has been successfully renewed and it is valid from August 2024 through August 2026. Okay, in my next slide here, these are the GLP services that we provide under NCRF IMR. We provide single dose or acute toxicity study following the OECD TG 420, repeated dose 28 days toxicity study, subacute study, OECD testing guideline 407, and also repeated dose 90 days toxicity study, also known as the subchronic study, following the OECD testing guideline 408. So when it depends on the test item or the product that the sponsor gives to us. We also incorporate other international guidelines if needed, by referring to example guidelines such as the ICH guidelines, WHO, US FDA, and also EME, guidelines from EME. We will now proceed with a short video presentation highlighting NCRF IMR. This video presentation provides an overview of our GLP infrastructure and operational processes implemented in NCRF IMR. Thank you for continuing to tune in. So I will move on to my next part of the presentation, that is the roles and responsibilities of sponsor, and I will also provide some insights on the challenges faced by them when submitting their test item to our test facility. So first of all, what is a sponsor? Okay, so a sponsor is an entity, okay, such as pharmaceutical companies, research organizations, universities, or even government agencies that commissions, supports, and submits preclinical safety studies to regulatory authorities. Sponsors are also the fund and resources provider in order for a GLP study to run until its completion. The OECD document number 11 was established to serve as a guide for stakeholders to assume the roles and responsibilities of a sponsor. So this document is available online. So all those who are interested to send in their final formulated test items to GLP compliant facilities, you may acquire this document from the website, OECD website, and look through and know and understand your responsibilities when submitting a product to a GLP compliant facility. Okay, so I have listed here the some key points of the roles and responsibilities of sponsor based on OECD document number 11. Okay, so first of all, sponsors are required to understand the requirements of principles of good laboratory practice, especially on the test facility management, quality assurance, and also the roles and responsibilities of a study director. Yeah, and sponsors also must ensure the test facility is able to conduct the study in compliance with GLP. One way to know is sponsors can also contact our NPRA, our regulatory authority, to know whether what are the test facilities that are available and also are certified to conduct the GLP study. Sponsor also must be aware of the content and also approve the study plan of the GLP study, and also to understand the content of the final report as well. All right, and a sponsor must be able to identify storage and retention of records and materials after the GLP study has completed. At least they must be aware that the GLP test facility has an archive to keep all their records and study records and materials. A sponsor also is responsible to submit all the data to regulatory authorities in order for reviewing and also approval of their product. And this point I put at the last here, characterization of test item or product is actually is the most important of all for sponsors, because sponsor and also test facility must be able to verify the identity of the test item subjected to the GLP study, so that there's no any mix-ups, or so that we are able to identify and verify the data once the GLP study has completed. All right. When sponsors are acquiring and submitting test items for a GLP study in NCRF IMR, they encounter various challenges that can affect the quality, compliance, and timelines of GLP study. Okay, so first of all, sponsor, the challenges that they face, is even by the characterization and the identification of the test item or the product. Sponsors must ensure the test items are properly characterized, be it in chemical composition, purity, structure, or biological activity, to meet the study protocol requirements. Inadequate characterization may may compromise the reliability of study results. Secondly, quality assurance of test item or product. Quality here can be, one example would be the consistency of each batch of the test item is essential, yes, for reproducibility and validity of the results. Variation between batches, if in terms of purity, composition, or even stability, can lead to unreliable results and non-compliance with GLP. Some sponsors face logistical issues, such as maybe on the shipping and handling of the test item itself. Another example would be inventory management, whereby inadequacy of the test item itself to inadequate test item submitted to a test facility to conduct the GLP study can be a problem too. Ensuring compliance with GLP regulations. GLP studies require comprehensive documentation for every step of the process, which include batch number documentation, certificate of analysis, COAs, and certificates of good manufacturing practice, yeah? All of which must be maintained meticulously to ensure traceability. And also other challenges faced by sponsor is the lack of communication between sponsor and the test facility. And also, GLP study can be very expensive. So because of the processes that's involved in it, so costing and cost management can be a challenge as well. And another basic challenge would be the awareness on the principles of GLP. Many sponsors do not know or have a lack of awareness on acquiring the principles of GLP. So how to overcome the challenges, okay? So therefore, proper planning, communication, and collaborations with reliable partners are critical in overcoming these challenges and ensuring successful study outcomes in a timely manner. Okay, so here I have provided a checklist, right? Whereby sponsors can refer to this checklist when you are planning to send in your test item or product to our facility, NCRF IMR. So of course, first of all, we would request for certificate of analysis of the test item or the product. So whereby it is best to have it GLP under the GLP from a GLP certified facility. But if you don't have that, it's okay. So as long as you have ISO 17025 accredited facility is also accepted. Okay. So the information that we need in the certificate of analysis would be such as your product name or even in a code name. Yeah. So batch number is also required. It is very important. And the composition, expiry date, and of course the storage condition of the product or the test item that you're submitting, as in the temperature range that it should be kept, whether it is sensitive to light or needs certain humidity range to be kept. Yeah. You must also clearly state the physical condition of the test item, okay, as in the appearance, the identification, color, order, and so on, okay. So in a certificate of analysis, you should have also the results from your microbial analysis that you conduct, and also heavy metal analysis. Possible if you have any other data, such as solubility and disintegration time, would be important too. Okay, so finally, the formulation. Okay, so the formulation must also be a final one. Okay, even if you're sending in for a preclinical toxicity study, because the same formulated test item or product will be used later in the clinical study in human trials later. Right, so the formulated product is final formulated product is really very important, yeah, for GLP preclinical toxicity study. And you also should state whether it is in capsule form, tablet, or in a liquid form, powder form, or in a sachet, and so on. All right, so there's also additional information that we will request as well from sponsors. That is the sponsor name and address, and even if you submitted your test item to a manufacturer to manufacture your product, we would need that information as well, the manufacturer name and address. If the test item need to be diluted, we would want the information of the vehicle used for the dilution of the test item. All right, and also other studies such as stability studies and the route of administration of your product, and so that we can apply it into the study animals, therapeutic dose, indication of use, and also information or information on previous efficacy studies that has been done on your product or your test item. Okay, and also other information such as transportation methods, labeling, and special instructions of your product is also very, very crucial for us to know, so that we're able to follow the same requirement when we are conducting the study and applying it on study animals. Okay, so that concludes my presentation for today. I hope everyone whom have tuned in have found my sharing to be valuable and beneficial. If you have any further questions or would like to discuss any points in more detail, please feel free to reach out to me via my email address. Yeah. Thank you all for your attention, and I wish you a wonderful day ahead. Back to you, Dr. Lau.
Okay. Thank you, Puan Suganthi, for those clear and actionable insights. So we will take questions after our second presentation. So please keep posting your questions via the Q&A link so that we can group similar topics together. So our second speaker is Dr. Nur Azra Mohamad Pauzi, a hospital pharmacist at Hospital Shah Alam, with a PhD in toxicology and prior experience as a regulatory pharmacist at the National Pharmaceutical Regulatory Agency, NPRA. So she will be presenting Pyrrolizidine alkaloids exposure in Malaysian natural products and herbal tea. Dr. Nur Azra, the screen is yours.
Thank you, Dr. Lau, for the kind introduction. Bismillahirrahmanirrahim. Allahumma salli ala Sayyidina Muhammad wa ala ali Sayyidina Muhammad. Assalamualaikum and a very good morning to everyone. My name is Nur Azra binti Mohamad Pauzi, and I am a pharmacist from Hospital Shah Alam. Thank you for joining today's webinar, and also thank you to IMR for providing this platform for me to share my research and my knowledge. Okay, so for today, I will be sharing on pyrrolizidine alkaloid exposure in Malaysian natural products and herbal tea. So let's start with a simple question. What are natural products? What do we mean by natural products? Well, in general, natural products contain natural substances such as plants and herbs as an ingredient, but there are a wide range of items. As you can see here, the Drug Registration Guidance Document, or DRGD, which is an official document provided by the NPRA, stated there are 5 categories of natural products. The first one is traditional medicine. Second is herbal products. Then we also have homeopathic medicine. And we also have natural products with modern claims and natural products with therapeutic claims. So the type of evidence that is required for each product category are different. If you would like to know further, you can always go to NPRA website and find this document. Okay, this table shows different product categories, their format of MAL number, and the product claims that they're allowed to make. For traditional medicine, herbal products, and homeopathic medicine, the MAL number ends with a T at the end, and the permissible claim is usually very general. Okay, it must start with traditionally used, and the claim is general, for example, to improve general health. Okay, for natural products with modern claim, the MAL numbers ends with an M at the end, and the claim that is used is more scientific but not too specific. Okay, for example, in here, herbal products used to support healthy cholesterol level. And for natural products with therapeutic claim, the end of the mile number ends with a B, and they are substance, okay, they contain substance or standardized extract, which is a known amount of the extract, and they are allowed to make specific claim. For example, herbal products used to reduce cholesterol levels. Okay. Okay, so natural products are not only popular in Malaysia, but they are also popular worldwide. Okay, all over the world are consuming natural products. We can see that nowadays people are moving towards natural products rather than conventional pharmaceutical medicine. Okay, why? Okay, number one is because more people actually believe that they are safe. They are always safe. And secondly, they are more affordable than conventional drugs, and also they are highly accessible. What does it mean by highly accessible? Well, number one, you do not need prescription in order to get traditional medicine or natural products. And secondly, you can even buy it everywhere. You can buy it on TikTok, you can buy it on Shopee, you can buy it online, you can even buy it from normal regular store. Okay. So some statistic here. Okay, 64% of Malaysians have reported the use of traditional and complementary medicines, and the WHO 2019 report also stated that 30% of Malaysians have used TCM at least once in their lifetime. And in fact, in Malaysia, natural product industry is a significant industry. In 2024, it was valued at US dollar 11.4 billion. However, natural doesn't always mean safe, okay? Natural products have their own issues. For example, they may be contaminated, they may have poor quality control, they may actually contain naturally occurring phytochemicals, and they may also be adulterated. Okay, in terms of contaminations, there have been many reports of natural products containing heavy metals such as arsenic and mercury and cadmium, microbial content, high microbial content, and also contain pesticide. And natural products also often suffers from poor quality control. And moreover, some of the herbs that's used in the natural products may contain naturally occurring phytochemicals, which can be hepatotoxic, which means they are dangerous to the liver, nephrotoxic, which means they are dangerous to the kidney, and they might also be genotoxic and carcinogenic, which means that they may cause cancer. Okay, some of these products also are known to be adulterated with either similar plant species, or they are adulterated with synthetic drug, which means that they are not supposed to be there. If you are taking a natural products for pain, it's not supposed to contain any pharmaceutical ingredients such as paracetamol or ibuprofen. So however, some of the manufacturers, they actually added this to potentiate the effect and also to increase the sales. So what does this mean? So while natural products can be beneficial for you, but you must not take the safety for granted. So always choose your natural products well. Okay, so one of the important group of natural toxins is the pyrrolizidine alkaloid, which is the focus of today's research. So what is pyrrolizidine alkaloids, or PAs? Okay, so they are a compound, they are a group of compounds produced by more than 6,000 plant species, particularly in the Asteraceae, Fabaceae and Boraginaceae families. And they are actually, the purpose of this compound in plant is to act as a defense mechanism against pests and also grazing animals. Okay, they are found in many different plants, and they are very unique because they actually exist in a wide range of congeners or different structures, and the toxicity and the potency of pyrrolizidine alkaloid molecule depends on the structure. However, which means that there are some which is non-toxic and some which are less toxic. However, there are three PAs which are widely studied, which are lasiocarpine, monocrotaline and bridalin. Okay, these compounds are actually categorized as 2B by the International Agency Research on Cancer, which means that they are potentially carcinogenic, or they can cause cancer. Okay, so here is the basic or the core structure of a PA molecule. So all PA molecule will have this structure. So they consist of two parts, which are the nesine base and also the nasic acid. So the nesine base, as you can see here, is a two-ring structure with a nitrogen atom in the middle, and it also has a double bond here. Okay, and connected to the nesine base is esters. Okay, the nasic acid 1 and nasic acid 2. However, this nasic acid can be either just one or two molecules, and sometimes can also be in a ring structure. And all this, the way the molecule is structured affects how it's metabolized in the body, therefore it affects the toxicity. Okay, so for now, there are more than 900 types of PAs that have been identified by scientists, and I will explain about the toxicity after this. Okay, as you can see here, okay, these are the highly toxic and more studied PAs compared to others, which are lasocapine, bridalin, and monocortalin. So upon ingestion, which is normally through diet, they will be metabolized in the liver, and then it's not always excreted through the urine, and instead it gets accumulated in the liver and cause toxicity reactions. And the toxicity occurs in the liver, so the symptoms are normally ascites, cirrhosis, liver enlargement, and veno-occlusive disease. Whereas these two PAs, platinacin type and PAN oxide, are less toxic than the other three, and upon metabolism in the body, they will be excreted in the urine. So over the years, outbreaks of PA poisonings have been reported all around the world. For example, in the 1920s, the first recorded intoxication of pyrrolizidine alkaloid, where people developed chronic liver disease symptoms from eating bread with wheat contaminated with PAs. And in the 1940s, in Central Asia, more than 50 people have died and 250 people were hospitalized after eating grains contaminated with PAs. And in 1970s and also in 2008, specifically in Gulran district, Afghanistan, the entire communities developed veno-occlusive disease from eating contaminated wheat. So beyond the outbreaks, studies have also shown that PA contamination occurs in many everyday products that we consume. Okay, for example, in Indonesia, a study was conducted whereby the scientists collected jamu samples from the market and found that 97% of the jamu samples were contaminated with PAs. And in China, 58% of the honey samples were contaminated with PAs. And in Europe, PFS, which is plant food supplement, 60% were contaminated with PAs, and in Ghana, herbal medicine, and also in Brazil. Okay, this shows how widespread this issue is. So the next question is, how about Malaysia? Okay. So with this background, my study was set out to find out or answer a key question. What is the health risk of PAs in Malaysian natural products and herbal tea? And we chose to answer this question by using a range of analytical, modeling, and risk assessment approach, or what we call integrated approach. So how was this conducted? Okay, number one, we identified the number of registered natural products in the Malaysian market that may contain pyrrolizidine alkaloid. And secondly, we quantified the level of pyrrolizidine alkaloids in Malaysian natural products and herbal tea samples that we have collected. And using that result, we assessed the health risk from consumption of those products using the margin of exposure approach of MOE. And then we predicted the acute toxicity of PA consumption using PBK modeling. And finally, we mapped and characterized the health risk of these products by using the risk 21 approach. So a little bit on how I conducted the experiment. So first of all, I obtained the samples, okay, that may contain PAs from online and also from physical store. And then the samples were processed. If they are not in powder form, then I ground into powder form, fine powder form actually. And then the samples were extracted in methanol. And after extraction, filtered into vials and then stored in -20 degrees until further analysis. And analysis was conducted using LCMS, which is a chromatographic method. And I would like to thank Mardi and also USM for providing me the platform to use the equipment for me to conduct the analysis. So what did we find from the study? Okay, number one, we have identified 114 registered products from the Quest research that may contain pyrrolizidine alkaloid, which means that these products, based on the ingredients, they contain plant species that are known to produce pyrrolizidine alkaloid. And then from the lab analysis, we found that 11 out of 30 samples or 37% were positive with pyrrolizidine alkaloid, with concentration ranging from 0.02 to 21.1 microgram per kilogram. And by using this result, we were able to calculate what is the estimated daily intake for Malaysian population, okay, specific for Malaysian population. So the intake, the estimated daily intake for pyrrolizidine alkaloid in Malaysian population is between 8.48 picogram to 1.6 microgram per kilogram body weight per day. Okay, so from all the positive samples, okay, we conducted risk analysis using, risk assessment using MOE approach. So 8 out of 11 positive samples had margin of exposure level, MOE level, below 10,000. And this is for for long-term consumption, which means what is the risk of developing hazard from consuming these products, okay, over 75 years every day. Okay, so it's below 10,000, which means that this product actually require risk management. However, for short-term intake between 2 weeks to 6 weeks, okay, all the MOE were above 10,000, so they require less risk management. Okay, we also conducted the risk 21. We also applied the risk 21 framework, which combines exposure data and hazard data to provide a visual risk map. And as you can see here, okay, so these for all the 11 positive products, the risk of developing hazard, okay, is actually ranging from low moderate to high moderate. So this is the risk 21 plot. Okay, so if the products actually falls in the green section here, it means that they are actually in the low risk, and in moving on to the red color, okay, is basically from low to yellow, and orange is moderate, and to red is high risk. Okay, by using in vitro, in vivo extrapolation, I was able to predict the benchmark dose, okay, that would cause toxicity in animals, okay. So in my study, we did not conduct any in vitro or in vivo study. However, it was important for us to understand how this concentration, you know, from the products that we have tested, how will this actually affect, okay, the organisms, okay. So we used in vitro studies, okay, to plot our in vitro concentration response curve. Okay, so in the left here, okay, this is just a representation. Okay, it's not the exact data that I have produced. Okay, so here we use the in vitro data for PAs and use the PBK model, which is physiologically based kinetic modeling, which is a computational model, to convert this or to extrapolate this into dose response curve. Okay, so this was conducted in cells, okay, and by using the PBK model, we were able to predict how it will actually behave, the chemicals will behave in animals. We can actually use the the models for animals such as rodents and also for animals without conducting the tests on the organism itself. Okay, so from this, we were able to predict that the BMD or benchmark dose, which is the dose that will cause toxicity reaction, and specifically for my study, we used rats, okay, as a model, and we found that it can range from 9 to 89 milligram per kilogram per day. So how does this relate back to the result that I found before? So if we see the concentration that we found, which is the estimated daily intake, which is much lower than the predicted BMD, so it means that it may cause, is lower than the level that is expected to cause harm in rats. So these advanced tools actually help us to predict the toxicity in humans more accurately, and even without extensive animal testing. And this is actually good for preclinical studies. And this is part of the new approach methodologies in toxicology. Okay, so to wrap up here, there are three main take-home messages that I would like to convey. Okay, first, some natural products are not as safe as we think. Long-term use can actually cause health risk if they are contaminated with toxic compound such as pyrrolizidine alkaloid, heavy metal, and others such as pesticide. And second, margin of exposure approach, PBK modeling, and risk 21 are very powerful tools that can actually help to assess the toxicity of a compound. And finally, there is also a real need for better monitoring, stronger regulation, and more public awareness for the safety of natural products and herbal medicines so that people can continue to use natural products in confidence. So I would also like to emphasize that our aim is not to stop people from using natural products, but instead we would like to educate people and to show that you are making a wise choice when choosing your natural product and to be vigilant in choosing your natural products. So to make sure that you are taking products that are safe and of good quality. Okay, that is all from me. If you would like to read more about my research, feel free to visit my ResearchGate and also my Google Scholar website for more papers. And thank you for your kind attention. Back to Dr. Lau.
Okay, thank you, Dr. Azra, for the insightful presentation. So we have come to this Q&A session. So please send your questions using the link in the YouTube description. So I will do my best to direct your questions to our distinguished speakers. So if we can't address every question today, we will invite the speakers to follow up by emails afterwards. So it looks like we have received a question from the audience. Please give me a second. So there's one question from Nurul Ain. So for Puan Suganthi, so what is your opinion on using human cell lines to replace animal models in toxicity studies, for example, in hepatotoxicity or cardiotoxicity testing? Considering that animals often differ significantly from humans, such as in heart rate, do you think human cell models could provide a more accurate alternative?
Okay, thank you. Thank you very much for your question, Nurul Ain. Okay, so, well, my opinion. Yes, in conducting preclinical toxicity studies, so as I said, there's in vitro studies and also in vivo studies. And yes, animals often differ significantly from humans, but according to the guidelines, yeah, all types of based on your product is essential to be conducted so that the data that's produced will be, you have the data. And also when you want to send in for your product registration, if it's required by our regulatory authority, a sponsor should be able to submit all of the data. Yeah. So, well, what do I think about human cell models, whether they can provide a more accurate alternative? Well, I would always go back to the guideline. Yeah. Okay. So whereas we have many methods out there, but what has been depicted in the guideline, we shouldn't forego or this one. So we must. It's advised to follow the guideline. We must be able to have all the data that's related to our product and submit accordingly to the regulatory authority. So that's my opinion. Thank you.
Okay. Thank you, Puan Suganthi. I hope, Nurul Ain, you got your answer. So we have one question for Dr. Azra. It's from E.T. So she asks, are there any visible symptoms or signs of Pyrrolizidine alkaloid toxicity?
Thank you, Dr. Lau, and thank you, E.T. PE toxicity affects the liver. They are highly hepatotoxic. So the effects the symptoms that will be shown is all related to the liver intoxication. For some of the symptoms will actually look like jaundice. You have a yellow, the turning of the skin and the eye, the white eye to yellow, and also you also have some of the patients will actually have ascites, okay, which means that the abdomen is enlarged. Those two are the main visible PA symptoms for PA intoxication.
Okay, thank you, Azra. Okay, so I hope E.T. you get your answer. Okay, the second question to Puan Suganthi, okay, from Dr. Siva. He asked, when a sponsor send a product for, let's say, 90 days toxicity analysis, will the work fully be carried out by GLP's own staff, or the sponsor needs to send the research assistant or officer to support the experiment?
Also, I would like to extend my thank you to Dr. Siva for the question. Yes, if it's for 90 days subchronic study, we do have our NCRF IMR consists of close to 50 personnel. Okay, so we will fully utilize our trained and competent NCRF personnel. But if you're asking whether sponsor can send their RA or officer in order to conduct the study, so I will always refer back to the guideline, same, yeah. So sponsor has the role of providing resources, right? So sponsors can send in their RAs and or officers to come in together with us in our facility to conduct the study. But before the start of the study, we will ensure the personnel or the officers undergoes our OECD-GLP training, and also some specific trainings such as going through our SOPs and also some of the procedures that's involved in the study, training first. Yeah. So, yeah, I hope I answered your question.
Okay, thank you, Puan Suganthi. So I hope Dr. Siva got your answer. If not, please come and meet Puan Suganthi in IMR. Okay. So we have last question to Dr. Azra. Is from Zairul. So he asked, what is the current monitoring tools used in Malaysia to assess the MP's toxicity? Is there any?
Thank you, Mr. Zarul, for your question. Okay, so prior to when a company submits their product for registration, they are required to submit the COA, which is the Certificate of Analysis, and the COA must also contain information on the contamination. For example, herbal metal, heavy metal contamination, arsenic, cadmium, mercury, and lead. Okay, they must contain the information on the levels of those 4 compounds, and moreover, they must also have the information on the microbial tests, so for example, MLT, microbial limit test on the Certificate of Analysis itself. So so that is one of the monitoring that is required during registration. And for post-registration, okay, companies must actually send their samples, okay, but I can't remember exactly how long or the period that they must send the samples after the registration. So the samples will then be tested for those 4 compounds, or also for microbes after the registration process. This is what we call post-marketing surveillance. Okay, and moreover, if anyone actually have any complaints about natural products, they can always send their sample to the program Perkhidmatan Pharmacy, especially the enforcement part, for us to then conduct the test on the toxicity.
Okay, thank you, Dr. Azra. So I hope Zairul got your answer. And okay, we have one last question. This is for Puan Suganthi. Okay, from I suppose is Dr. To from hospital. So he asked, or she asked, may I know what are the projects or non-clinical studies have been conducted in your GLP facilities for the past 2 to 3 years, and what is the process like? What are the process like if we would like to propose a project or study in GLP IMR?
Thank you again for the question, To. All right, what are the previous projects or preclinical studies that we have conducted before this? Yes, our services, we provide general toxicity studies. That is the acute toxicity study and repeated dose toxicity study, 28 days and also 90 days. So for the past 2 to 3 years, our experience conducting the GLP study in our facility would be the acute toxicity study. But we have performed 28 subacute study before, and also 90 days subchronic toxicity study as well. So what would the process be like? Just write into us, okay, so via email. And because some other facilities, other GLP test facilities, they have provide forms and all that. But for us, you just have to write into us. That's all.
Okay, thank you, Puan Suganthi. So I believe Dr. To have got your answer. So I'm afraid that's all the time we have for Q&A. So any additional questions will be shared with our speakers for follow-up by email. So we hope today's webinar has shed light on current research at IMR and sparked interest in future collaborations. So before we conclude, I'm pleased to announce our next IMR-SEAMEO Webinar Series session on 31st of October 2025. So please mark your calendars. And the theme is Decoding Rare Disease Using Bioinformatics. So we hope you can join us at the next session. Until then, many thanks from all of us at IMR. Take care and stay safe.